In this video, we are going to learn about bile, how bile is produced by the liver and then it is stored in the gallbladder. We are briefly going to go over the anatomy of the liver and then we will talk a little bit about bile. So there are three accessory organs which secrete their secretions in the alimentary canal. They are liver and gallbladder, and pancreas. Liver, its function is to make the bile, and that bile, it is a fat emulsifier. Just like the detergent, it emulsifies the fat. The bile, it emulsifies. It also emulsifies the fat, and that bile it is stored in the gallbladder. And then we have pancreas, and as you already have learned, that pancreas it secretes many enzymes into the alimentary canal, and it also secretes bicarbonate to neutralize the stomach acid. Now let's go to the liver. So liver, it is a large gland. It consists of four primary lobes: right lobe, which is the largest, and the left lobe, caudate lobe, and a quadrate lobe. This caudate lobe and quadrate lobe, they are present on the inferior surface. Then its mesentery is a falciform ligament. It separates the large upper and the small left lobes, and it suspends the liver from the diaphragm and the interior abdominal wall. So this is falciform ligament. This is mesentery of the liver. Remember, we talked about that the two peritoneum layers, they come together and they form the mesentery. And the mesentery, it helps the organs to attach to the abdominal wall. And then the lower part of this falciform ligament is known as round ligament. It was umbilical vein in the fetal life. This is liver. This is the right lobe. This is the left lobe. And this is our this is quadrate lobe and this is caudate lobe. This area of the liver, it is known as bare area because it is not covered by peritoneum. We know like visceral peritoneum, it covers the outer surface of the organs, but this area of the liver, it is bare area. This is gallbladder. So the bile, it is produced in the liver and then it is stored in the gallbladder. Then liver is attached to the stomach through lesser omentum. We talked about this when we were doing the stomach as well. And in the lesser omentum, the hepatic artery and the hepatic vein, they travel and they reach the porta hepatis and also the bile ducts, which are the common hepatic duct. Common hepatic duct, they also travel in the lesser omentum. And these common hepatic ducts, they leave the liver and then they join the cystic duct to make the bile duct. So the common hepatic ducts, they join the cystic duct to make the bile duct. This is the diagrammatic form. This is the right lobe. This is the bare area of the right lobe. This is the left lobe. This is the caudate lobe, and this is the quadrate lobe. And here you can see the gallbladder. They made it green. And then if you look, this is the bile. This is the bile duct. This is the cystic duct, and this present inside. This is the common hepatic duct. So common hepatic duct and cystic duct, they join to form the bile duct. Now, microscopically, liver, it is made up of hexagonal lobules, and it is composed of plates of hepatocytes which surround a central vein. And then there is a portal triad in each corner of the lobule. That portal triad, it contains a branch of hepatic artery, hepatic portal vein, and a bile duct which receives bile from the bile canaliculi. Let's see. So these are hepatic lobules and they are separated by the connective tissue. This light area in the middle, this is central canal, central vein. This is a closer look, and this is a central vein around which there are sheets of hepatocytes are present, and between these hepatocytes there are dilated capillaries which are known as sinusoids. Okay. So the liver sinusoids, they are leaky capillaries located between the hepatic plates. Blood from both the hepatic portal vein and hepatic artery proper, it percolates from the triad region through sinusoids and empties into the central vein. And in these liver sinusoids, macrophages are present. They are known as hepatic macrophages. Now let's look at this. It will make it clear. This is a liver lobule, right? And on all the six corners, this is portal triad is present, which consists of a hepatic artery, a portal vein, and a bile duct, right? And then these are the hepatocytes, the hepatic plates, which are surrounding the central vein. These hepatic plates. Between these hepatocytes, we have dilated capillaries, which are known as sinusoids. So blood from this vessel, from the artery and the vein, together, both of them, they mix in the sinusoid. They move in the sinusoid, and then they go to the central vein. Whereas the bile, it is produced by the liver cells and it moves in the opposite direction towards the bile duct. So blood is moving this way, the bile is moving this way, and then these bile ducts, they join to form the hepatic duct, which joins the cystic duct to form the bile duct. Now, hepatocytes, they have increased rough endoplasmic reticulum and smooth endoplasmic reticulum, because liver, it synthesizes many compounds, and then it also detoxifies many compounds. So liver cells, they are rich in as a rough and smooth endoplasmic reticulum, Golgi apparatus, peroxisomes, and mitochondria. And what are the functions of the hepatocyte? They produce almost 900 milliliters of bile per day, and then they also process the blood-borne nutrients. The blood from the gut, it comes to the liver. And how do they process the nutrients? For example, they store glucose as glycogen and make plasma proteins. And they also store fat soluble vitamins, and they also perform detoxification, for example converting ammonia to urea. So liver cells, they are very important, and they are doing many functions in the body: producing bile, processing the nutrients, storing fat soluble vitamins, and then they also perform detoxification. Now let's come to the bile. Bile, it is a yellow-green alkaline solution. It contains bile salt and bilirubin. Bile salts, they are cholesterol derivatives, and they function to emulsify the fat, and they also help in the absorption of the fat. So when we went over the fat absorption, we discussed, let me just quickly remind you. So the fat is coming in the gut lumen, in the alimentary tract, and then the liver, it secretes the bile, which the liver creates the bile, stores it in the gallbladder, and when there is need, that bile, it is pushed into the gut, and that bile, it attaches to the fat present in the food. So the big fat molecules, we know that the fat molecules, they tend to come together when they are in aqueous solution, because in the alimentary canal there is the water base, the chyme is water based. So the fat, it comes together and it forms big molecules. So the bile salt, they act on it just like a detergent acts on a fat, and they emulsify it. They break down those big fat lobules into small lobules, and those lobules, and enzymes from the pancreas, they act on those small lobules. They cannot act on the big lobules, and then those are simplified, those are made into uh, simpler fat form, and that simpler fat form is again, it attaches to the bile and it makes micelles. Those micelles, they are taken to the apical border, and in the apical border, the fat, it gets off from the micelle, it gets off from the bile salt, and it enters the cell. And inside that cell, the gut cell, it is then made into chylomicron, and chylomicron is secreted into the lacteal system. All right, so this is about bilirubin, so about the bile salt we were talking. So what is bile salt doing? They're emulsifying the fat and making the absorption of fat possible. Then another component of the bile is the bilirubin. It is a pigment formed from the heme. Bacteria, they break down in the intestine to stercobilin and give the brown color to the feces. This bilirubin is responsible for the brown color of the feces because there are bacteria present which break down this compound. And if you remember when we were doing blood, we said that the heme portion, it is turned into bilirubin. So this the bilirubin is a component of the bile. And then it has cholesterol, triglycerides, phospholipids, and electrolytes. So these are the components of bile: bile salts, bilirubin, cholesterol, triglycerides, phospholipids, and electrolytes. Now let's see. So there is enterohepatic circulation. Entero means intestines and hepatic circulation. And there is recycling mechanism that conserves the bile salts. So the bile salts, which are the bile which is secreted into the gut lumen, it acts, and the bile salts, they act on the fat. After that, the bile salts, they are reabsorbed. Bile salts are reabsorbed into the blood by the ileum. Ileum is the last part of the small intestine, and they are returned to the liver via hepatic portal blood, and they are resecreted in the newly formed bile. About 91% of the 95% of the secreted bile salts are recycled, so only 5% is newly synthesized each time. Now let's go over this. This is our liver and this is the gallbladder. Bile, it is produced in the liver, it is stored in the gallbladder, and then we know that the hepatic bile duct, it joins the cystic duct coming from gallbladder, and they make the bile duct. That bile duct, it secretes the bile in the duodenum, and when the food comes, that bile, the bile salts, they emulsify the fat and they cause the absorption of fat possible. And then these bile salts, they are reabsorbed in the hepatic portal vein, and they are taken back to the liver and they are secreted, they're again incorporated into the bile. So this is how bile salts, they are salvaged. What can go wrong with the liver? So we all know hepatitis, it is viral infection of the liver. It can also be due to drug toxicity and due to a mushroom poisoning. Then cirrhosis, chronic hepatitis, it can lead to cirrhosis, or alcoholism can also lead to cirrhosis. In cirrhosis, there is fatty fibrous change in the liver, and there is portal hypertension. Then portal hypertension has its own symptoms through which we can detect that this person's liver is going into cirrhosis. Then liver transplants are successful, but they are very rare. Liver can regenerate to its full size in 6 to 12 months after 80% removal. So this is very interesting fact about liver. That is. And now I said that bile is synthesized by the liver and stored in the gallbladder. Gallbladder is a thin-walled muscular sac on the ventral means anterior surface of the liver. It functions to store and concentrate bile by absorbing water and ions. So when bile is stored in the gallbladder, it absorbs the water and ions, and this way it concentrates the bile, and it contains many honeycomb folds that allows to expand as it fills. And muscular contractions, they release bile via cystic duct, which flows into the bile duct. So this is something about gallbladder, and some especially in women, in fat women, gallbladder can get inflamed, and sometimes it can have stones and it needs to be removed. So that is all about bile.